Cool. Thank you everyone for turning up today. My name is Raylea, as you heard, and I'm going to give you an introduction as to what I'm studying my PhD on. The title is Environmental Ecology and Raylea Virus Vector Aedes camptorhynchus here in Tasmania. Got two local supervisors, Jeff and Leon, who are both present today, and two non-local supervisors, Phil and Scott. So what is Raylea virus? Raylea virus is a mosquito-transmitted disease. It's endemic to Australia and the Pacific Islands. It was first identified in Australia in 1958 and in Tasmania in 1981. It presents severe joint pain, rash, fatigue and prolonged morbidity. There are 35 species of mosquito involved in transmitting Raylea virus. They represent over 35 families of mosquitoes. And there is a mammalian reservoir host, mostly kangaroos. In Australia, the primary vector for Raylea virus is Aedes vigilax, as you can see by the red dots. It's widely distributed throughout Australia, but in the southern areas of Australia, Aedes vigilax and Aedes camptorhynchus coexist, where in Tasmania, Aedes camptorhynchus is the sole vector for the virus. From this chart here, over the past 5 years, you can see that the majority of the cases occur in the warmer areas of the country, and it's quite sporadic throughout the years and throughout the states. This graph here is showing you the notification rate of Raylea virus per 100,000 population from 2000 to 2012. 2012 data is not complete yet because we haven't finished the year. And as you can see, that there is quite a high correlation between outbreak years between Tasmania in the blue and New South Wales in the red. New South Wales, on average, equate to 18% of Raylea virus cases in Australia. So you might ask what's creating these peaks and troughs, and most studies show that Raylea virus and outbreaks of mosquitoes are correlated with high rainfall, high temperature. But a lot of the cases that are reported throughout Australia are based on Aedes vigilax. So what I aim to do is look at environmental modelling of Aedes camptorhynchus here in Tasmania. This is going to be in collaboration with the Sorrel Council, as they have a high need for a predictive model for their early warning systems of Raylea virus outbreaks in Tasmania. The second part of my project is looking at competitor interactions between Aedes camptorhynchus and invertebrates within their environment, which I'll elaborate on later. I'm looking at oviposition preference of the mosquito in their habitat here in the state and hatching triggers of their eggs. Late last year, I selected 3 salt marshes in southern Tasmania. We've got Primrose Sands on the Carlton River, Cambridge and Richmond salt marshes on the Cole River. What I did was selected 10 water bodies, typical of the one down there on the bottom of the screen. All salt marshes are relatively similar in their density of camptorhynchus, their human impact in the surrounding area. And what I've done is selected 10 water bodies in each of those salt marshes to serial sample throughout the summer period. These serial sampling consist of dimensions of the water body, depth of the water body, percent cover of any algae and grass and samphire in around surrounding regions. They also consist of 5 larval dips using a 350mm dipper taken from majority of the habitats within each water body. From each of these dips, which are roughly 20 weeks, so 3,000 vials of invertebrates, I will put them into a quartered petri dish to subsample them, because otherwise it'll take me forever. And I'm identifying most of the crustaceans to order, such as amphipods, cladocerians, copepods and ostracods. Non-Culicidae flies are identified to family, which I won't attempt to pronounce, and all mosquitoes are identified to species. Other environmental parameters that are measured are rainfall, which are read on a weekly basis through that summer sampling period, and that is set up at each salt marsh, and they are correlated to the Bureau of Met data. I take air temperature using the Stevenson screens in the top right, and in there is a little eye button, a little silver thing there that records the temperature every hour. I also take parameters within the water body such as the salinity, temperature, conductivity and pH, and I'm also measuring the tide fluctuations within each salt marsh. Originally I was looking at the published data, but that was quite incorrect for the regions I'm looking at due to the river system that they're on, and the high local variation with moon cycles and things like that. So I've now deployed some tidal gauges out there to measure the tide. All of this information is going to be collaborated into a suspected and infected model, which has all of your larval stages, 1 through to 4, the maturity rate from larval 1 through to 4, the influence of competition, the death rate, and environmental impacts. You have a pupal stage, which has the same parameters, and the adult stage is broken up into susceptible and infected adults, and their contact rate, which is beta down here, is derived from published data. The second stage is looking at competition. A lot of studies have found that in high competition environments, larvae of various different species undergo an increased developmental stage, and there's a bit of discrepancy as to whether or not this influence increases the adult size or decreases the adult size. And if it decreases the adult size, you have a reduction in fecundity, but if it increases the adult size, it has been shown that the adults then can pick up the virus more readily and blood feed more frequently. So I was fortunate enough to be part of a preliminary study conducted with one of my supervisors here, and that looked at abiotic and biotic influences with camptorhynchus here in Tasmania. And what we found was that the ostracod here in the graph, you can see that camptorhynchus is on the y-axis and ostracods are on the x-axis, and you can see that there's a negative relationship between ostracod abundance and camptorhynchus abundance. So I'm going to investigate this interaction a bit further with my competition experiments. What I've done so far is set up some lab trials. As you can see, the first instar mosquitoes are quite small, as are the ostracods. And what I've done is put 50 first instar mosquito larvae into each of 40 containers, and then subject them to different levels of competitor densities, 0, 150, 300 and 600 ostracods in different treatments. And then run that through the whole life stage of the larvae right through to adult emergence. And I've recorded the survival, the stages it's taken, time it's taken for each stage of larval development, and then I'll be looking at adult wing sites, as that's a known parameter for adult size, and it's resulted in quite a lot of counting. Third stage is looking at overposition sites within the salt marsh. I've only chosen one salt marsh, which is the Primrose Salt Marsh, with its connection to the Sorel Council. They've done quite a lot of regular monitoring at this site, and there's quite a substantial number of mosquito larvae in this salt marsh. And what I've done is divided the salt marsh into 4 broad habitat types. You've got channels or tidal inlets, which were artificially put into the salt marsh in the 1950s. I've got salt pans, which are bare areas of highly saline sand. This one here is a samphire, which is a very low, dense vegetation. And then you have the salt bush on the other side. What I did, you can see here, this is an illustration of the salt marsh, the primrose sands, and the black areas were not sampled. And I've run a grid looking at high tide, medium and low tide, and gridded that out so that each grid contains one of each of the 4 habitat types. And what I've done is randomly allocated a grid in each of those sections and taken 3 soil cores from each habitat type, which will be repeated 2 more times next summer. And I've taken soil cores from each of those habitat types, looked at the humidity and the temperature in those microclimates, and then brought the soil cores back to the lab and inundated them with saline water for a period of days to look at what emerges from those cores, strained it, counted what's come out, let them dry again and re-inundated them, purely because Aedes eggs are known for installment hatching. This data is still being processed, and I'm yet to collect more to come up with a solid answer, but to date it looks like the predominant habitat type for Aedes eggs is the sandfly, which is what we predicted. The final stage of my experiment is probably the trickiest. It's looking at hatching triggers of Aedes camptorhynchus eggs. There's only been one person successful so far to rear them in the lab and get eggs from them to run any trials on them. So what I propose to do is collect adult females from the field using these adult traps, which contain dry ice in the bucket at the top, which releases carbon dioxide out of the tube at the bottom. The battery pack at the top supplies power to the circuit board, which runs a light and a fan in this white container here. And mosquitoes are attracted to the light and the carbon dioxide, and then they get sucked into the catching bucket at the bottom. And if the battery stops or for some reason fails, the trap is shut off permanently due to no air pressure. And the adults will be brought back to the lab and identified. The females will be put aside and bred in cages in a controlled temperature room and fed defibrinated horse blood, and then I'll hope to collect their eggs in ovi traps. These eggs will then be subject to different saline treatments and rainwater treatments to look at which natural environmental trigger is the best for hatching these eggs, so that they can complement the environmental model and the tidal predictions of each salt marsh. And I'll be looking at mortality rates and hatching time rates for the eggs. This project has been funded by the Holsworth Research Fund, which I'm grateful for, and I'd like to thank you for your time.